Type II hypersensitivity. Type II hypersensitivity is also referred to as antibody-dependent antibody-dependent cell cytotoxicity or cellular cytotoxicity. So of course, this one we've come across it before when we're discussing other aspects. So antibody-dependent involves so this is cell death where antibodies are attached to surface antigens, leading to cellular destruction. And mechanisms of this cell death happens by, of course, antibodies, where in this case we have IgE mostly, together with, of course, complement C3b. We know that C3b is an opsonin to a big extent, taken up by phagocytes. So complement is playing a key role in this, especially via C3b. This is one of the opsonins. The other opsonin, of course, is usually C4b. C4b is another opsonin. So ADCC happens in that the cells that are meant for death are coated with IgG. IgG coats those cells. In addition to IgG, of course, C3b can also coat those cells. And so neutrophils, which are referred to as polymorphs, monocytes, and even natural killer cells are able to target these cells because they're usually coated with IgG and or other immune complexes. So this ADCC happens in some reactions, including transfusions, when there is a mismatch. Of course, if somebody says blood group A, now umepeana blood group ya, umepeana your blood to somebody who's the same type of blood group, you're not a universal recipient, then what is going to happen is that there will be a mismatch, and the mismatch will happen so that there is a transfusion reaction, and this transfusion reaction will lead to the death of the red blood cells, and that happens because they are coated with IgG and, of course, C3b. And so once that happens, then they are marked by natural killer cells. Even cytotoxic T cells are able to kill these cells because they've been marked or they've been opsonized. The hemolytic disease of the newborn, we know that mother is rhesus negative, probably the child is rhesus positive. We know that the initial birth is okay. The initial birth is okay. These individuals are not affected. But what happens is that then the immune system of the mother is triggered. It is primed. So antibodies against rhesus positive are generated. So subsequent pregnancies, of course, will have issues. Yeah, so that is also referred to as the hemolytic disease of the newborn. So the newborn, of course, will be killed subsequently with antibodies against the rhesus. If the mother is rhesus negative and the father, the child is rhesus positive. How do you go solving such births? What happens to solve the problem? In antibody-mediated rejection, of course, of grafts, is the same thing that is happening. ADCC is happening there. Autoimmune disorders like SLE, rheumatoid arthritis, of course, the same is happening. The erythrocytes and platelets, of course, they will trigger this such immune response. So what is happening in type 2 hypersensitivity is that IgG, C3b bind them and the cell is killed. And of course, in that happening, it means that the cells are destroyed. So it's called antibody-dependent cell cytotoxicity as type 2. That is exactly what is happening here. This is what we're showing. So C3 is broken down to C3A and C3B, and C3B is an opsonin. So of course, once then complement is activated, ultimately we know that complement will bring about MAC, and MAC will bring about lysis of the cell. Similarly, IgG coats the cells, and the cells, once they are coated with IgG, then polymorphs, that is neutrophils, even macrophages and monocytes and natural killer cells know that either we are phagocytosing this cell or we are leading this cell to lysis. So there are a number of things that can happen, of course, during this type 2 hypersensitivity. So complement can cause lysis. the FC gamma receptor, of course, can bind to C3b. They bind to each other, and this leads to phagocytosis. That is another mechanism. Alternatively, we just have ADCC directly, where natural killer cells as well as myeloid cells kill those that are coated with IgG. That happens. Now, there's a question that I've asked you. Of course, we know that mismatched blood, we say the reaction is happening because of mismatch. So when you're doing mismatch, of course, an individual might suffer death because of destruction of the red blood cells because they become coated. But for the hemolytic disease of the newborn, how you solve that is that first of all, of course, the antigen is an isoimmune, which means the rhesus antigen, antibodies will be produced against it. If the child is rhesus positive and if the mother is rhesus negative, antibodies are going to be produced against it. But by the time they're being mounted, of course, for the first birth, the antibodies are not so many, so they won't even have any effect on the child. Subsequently, the IgGs against the rhesus have built up and they've been stored, of course. So subsequent pregnancies will be affected. So how you go about treating these individuals is that you're going to inject mothers with low doses of IgG anti-D, okay? So anti-rhesus factors IgG, so that these are antibodies against anti-D. So they are antibodies against antibodies. These are monoclonal antibodies. So that is what is happening. So you produce, you are going to inject them with IgG that is against the anti-D. So these are antibodies against antibodies. And see, since the good thing is that antibodies are proteinous in nature, we can always produce antibodies against antibodies. And that, of course, neutralizes those antibodies. So it's treating the antibody as an antigen. And that then, of course, subsequent pregnancies can be successful. And that is how you're able to deal with the hemolytic disease of the newborn. So that is type 2, ADCC. Type 2 is ADCC. Key factors there is either you're using complement that will form MAC, or IgGs coating cells, and these cells are going to be killed by immune cells.